So our next topic, now that we have a better understanding of how DNA works, is mutation. Now ultimately, a mutation is any change in the DNA sequence. So of course, when you think of mutation, you think of mutants, right? This cow, or the steer, is the result of a mutation. And that mutation results in doubling the muscle mass of this organism. Change in the DNA changes a protein, which changes a trait. Mutation. Sickle cell disease. So this is an example that we talked about earlier, and that is the idea that I've changed one nucleotide in the sequence of a gene for hemoglobin. Now that changes the hemoglobin such that it results in these sickle cells. It's a whole disease. So again, this idea that mutation changes the DNA. DNA encodes for the directions how to make a protein, the primary sequence of the primary structure of a protein, and that protein then is dependent on its form, shape, to do its function. And if it doesn't have the right shape, it doesn't do its function well, or it might do it better. So as we start to talk about different kinds of mutations, there are all kinds. There are big mutations and little mutations, what we call gross mutations. We have large chunks of chromosomes that are either rearranged or deleted or added. For now, what we're going to do is focus on what are called point mutations, a change in one or a few nucleotides. It's just one point in the DNA, so a point mutation. So two classes of point mutations, base substitutions and frame shift mutations. So a base substitution is just that. I'm going to substitute one of the nucleotides for a different one. Now, depending on what is substituted dictates the impact of the mutation. So because, again, we understand the dogma of molecular biology, we can see up the top of this slide what is called the wild type gene. And the wild type gene is any gene that is, in fact, the most common that you find in a population, the most common version. So we would quote-unquote call this normal, though there's no such thing because this is changing based on interaction with the environment, and that's evolution. But we'll talk about that later. Now, the wild type then in this case shows you 4 amino acids and the stop codon, it's the end of the protein. Now, in the first example, in a base pair substitution, we see that instead of GGC for glycine, we see GGU. So a U instead of a C. Well, because of the redundancy or the degeneracy of the genetic code, GGU happens to encode for glycine as well. So this is what we call a silent mutation. You've changed the DNA, but it hasn't resulted in a change in the protein. Now, a missense mutation, substitution mutation, is in this case we have an A instead of a G. So instead of GGC, we have AGC. And AGC doesn't encode for glycine, it encodes for serine. Now, changing that glycine to a serine may or may not have a big impact on the protein. You don't know until you make the protein and see how it acts, what its activity is like. It may be less effective, it may be not functional at all, or it might actually be more effective. It may be a higher quality enzyme for doing the job it needs to get done, and then it will be selected for by evolution, and this will then become more and more prominent because it does its job better. And ultimately, it could become the wild type. Now, if you get a nonsense mutation, so this could happen anywhere. In this case, it happens in the first amino acid, the second codon. It became a UAG. So instead of AAG, we get UAG, and UAG is one of the stop codons. And so all of a sudden now, when this gene is transcribed, you get a full mRNA molecule, but when you try to translate it, it stops immediately. So in a sense, this protein now has been deleted. Right? It's not going to work at all, and that certainly is going to have an impact on the organism. So our first category of point mutations are base substitutions. Right? So we change a single nucleotide. Now the second type of point mutation is a frame shift mutation. And this is a little bit trickier, but is potential to have a greater impact on the protein. So now what I'm going to do is insert or delete a nucleotide. Now when I do that, I shift all of the nucleotides downstream from that insertion or deletion, one direction or the other. All right. So, for example, here we're going to get a deletion effect in this frame shift causing extensive missense. So again, it's the same wild-type protein, but instead of the UUU for that third amino acid phenylalanine, we delete one of the U's. Now, there isn't a gap in the DNA. You don't have a blank space the way these deletions work. Everything gets shifted down one. So the G that was the first base for the codon for glycine now becomes the third base in the codon for the previous, for what used to be phenylalanine. Now, UUG is actually encoding for leucine. Now, if it was just a change of a single amino acid, it may or may not have an impact on the protein. But because you shifted everything down, now all of a sudden, what was GGC before, everything gets shifted down one. Now it's GCU because U was one more down from the C. And so GCU now encodes for alanine, and you effectively erased the stop codon because you shifted reading frames. Now, the reading frame is the idea that I'm reading the DNA in groups of 3. But those groups of 3 depend on where you start. So starting with AUG, methionine sets up 1, 2, 3, 1, 2, 3, 1, 2, 3, right? But if for some reason I started with UGA, now I have a different reading frame. There's different information by reading those groups of 3. So if I went UGA, AGU, UUG, GCU, got a different protein. And so then there's a third reading frame if I started with a G in the wild type, GAA, GUU, UGG, right? And then if I get back to A, so AUGA, if I start with A, and now I'm in the original reading frame. So what this says is that every strand of DNA actually has 3 reading frames, 3 layers of information. And that's another challenge then to accessing that information. I have to be in the right reading frame to get the right protein out of it. So you can see here is when we remove or insert another nucleotide in the second example, frame shift causing immediate nonsense, a stop codon is formed. Or if you lose 3 nucleotides, you may remove a whole codon and you maintain the same reading frame, but you've lost a whole amino acid or any combination thereof. Right? So frame shift mutations are when I insert or delete and I shift the reading frame of that mRNA molecule, what produces the mRNA molecule, everything downstream of that mutation event. All right? So those are point mutations. Now, mutation is natural and it's spontaneous. It's necessary for evolution to work. So all the diversity that we see is the result of mutants. You are a mutant, right? If you weren't, you would look like everybody else, right? So the different kinds of apples that you eat, the different kind of flies that are out there, the different people, the fact that you have different genes, different combinations of genes as a result of sexual reproduction, but the fact that there are different genes that can be mixed up and combined, all those different genes are actually the result of original mutation. Okay? And we'll talk about this more as we get more into genetics. But again, that idea that mutation is a change in the DNA sequence is really, really important to understand. Now, understanding mutation and change, the effect that a change in the DNA can have, we can talk about biotechnology. Because biotechnology is basically just a directed manipulation of that DNA in a modern sense of the word of biotechnology. So we know that DNA is copied into RNA, is translated into a protein. That protein does stuff that affects the trait. If I go back and change the DNA, I can ultimately change the trait, right? So if we talk about biotechnology in a very general sense, we can use the definition, the use of living organisms to solve problems and make useful products, right? And this definition is very broad and goes back about 13,000 years. Basically, when we first started doing agriculture and herding animals, we're using living organisms to solve problems and make useful products. Those products might be cheese or bread or beer or wine, right? So biotechnology, bio-life, technology, the application of science for the creation of products for human use, this is a very, very old concept. Now, most of the time in the modern world, when people talk about biotechnology, they're talking about a more modern definition. And this is the manipulation of living organisms at a molecular genetic level to produce useful products. And of course, this leads to lots of controversy because it is incredibly powerful. Right? So this is something that we can discuss more, the controversy of it, and in fact, you can get your whole degree in biotechnology. You can get your whole degree in bioethics. So again, we just want to introduce the concept so that you're aware of it and you can educate yourself more on it. But again, the idea is that if I take DNA from any given organism and I change it, I change that organism. And here, for example, I can take a gene of interest from one organism on the right and put it into a bacterium, in this case using a small circular piece of DNA called a plasmid. Then that gene, if it's expressed, can give new characteristics to the organism, such as the ability to clean up toxic waste, or I can use it to insert it into a different organism, like a corn plant. So that corn plant produces its own resistant pesticides, right? So now when a bug bites into it, it gets a mouthful of pesticide, it dies. Plant makes its own pesticides. Or I can use those bacteria to produce proteins that are hormones, or they're proteins that are therapeutics, that are you can use as drugs, very powerful drugs, because they're proteins. They can be enzymatic. So this whole manipulation on a molecular level, this is biotechnology. And because we understand the dogma of molecular biology, we can understand how to do this. And to be honest with you, it's not that hard. Right? Now, some people cry, this is unnatural. This is not something that was ever intended to be done. Like, we shouldn't be doing this. Well, we learned how to do this from nature. So there are bacteria out there, and one that I want you to know is called Agrobacterium tumefaciens. This is a very famous bacteria because this bacteria actually does biotechnology on a molecular level. So what it does is it inserts its own genes into a plant that it invades. And when it does that, it creates what's called a crown gall. Right? So you may have actually seen these on trees and plants and not realize what you're looking at, but it's kind of like a controlled cancer in that the bacterium, as it invades, it transforms. That means it's inserting its genetic material into the cells around it, into the plant cells around it, and changing them. And then those plant cells get the information that they're supposed to grow and form this tumor where the bacteria can live. And then they produce a bunch of very unusual amino acids that only the bacteria can metabolize to use for food. So food and shelter is being produced by the plant for the bacterium because the bacterium, Agrobacterium tumefaciens, is able to modify on a genetic level its host. Pretty crazy stuff. Very cool. So what we do is piggyback on that technology, on the ability of that bacteria to do that. And so what we do is modify the bacteria and say, instead of putting the genes you want bacteria into the plant, we're going to put the genes that we want to put into a plant and then let you infect the plants. And again, this isn't very hard to do. We could do this in any given lab that you could set up relatively easily. So that's plant transformation. That's biotechnology, and it's easier to do in plants than it is in animals, but that is coming along very, very quickly. And transformation of animal cells is quite robust now. And so instead of using a bacteria, though, to infect an animal cell, you use a virus. Because viruses can infect animal cells, as well as plant cells for that matter, and insert their DNA into the genome of the animal cell and transform it. And we're going to talk about viruses in our next recording, but they're a very, very important tool in biotechnology. And that's going to be our segue. Now, your last assignment is to click on this link, and I'll put the link on the website as well, and watch this talk. It's a relatively old talk. It's 5 years old now, or longer, depending on when you're listening to this. And but it gives you kind of an overview of some of the capabilities of biotechnology and asks you to think about them.